Hello everyone, I'm Edson Vieira. I'm a postdoc fellow at Marine College Lab at the Federal University of Rio Grande do Norte in Brazil. This work is a collaboration with Jessica Bleuel and Professor Guilherme Longo, and I'll show you a comparison between 2D and 3D data obtained from coral colonies. Although reef habitats correspond to only 1% or less of the oceanic surface, 25% of the marine species live in or depend on reefs for at least part of their lives. This high diversity is related to the complexity that corals add to reef surface, providing several habitats for feeding, mating, and refuging from predators. However, these sites are under several sources of threats. Locally, reef habitats are being affected by the decrease of fish abundance and diversity as a result of overfishing. Tourism is imposing physical damage to the corals. Besides, sewage, discharge, and oil spills are bringing chemical pollution, and even trash can accumulate on reef sites. And they are also impacted by global drivers. With the increase of the concentration of carbon dioxide in the atmosphere, oceans are changing fast and globally. The water is getting warmer, which makes corals to lose their symbionts and bleach, and also more acid, which affect the absorption of ions carbonate, resulting in the weakening of coral skeleton. So both warming and acidification is then resulting in coral mortality all around the world. When alive, coral reefs sustain a huge diversity. However, with the increase of global impacts and the consequent death of corals, reefs are becoming less complex and therefore not suitable anymore for several reef-associated species. Therefore, monitoring these habitats may help to understand how corals grow, reproduce, withstand, and survive, and also how complexity and diversity is maintained. Therefore, reef monitoring is crucial to understand reef functioning and how such habitats are dealing with local and global sources of stress. But here we have a problem, a huge problem actually. Traditionally, coral reefs are being monitored by data collected from photos that were taken in the field. However, corals are three-dimensional organisms and photographers usually flatten their complexity, precluding an entire analysis of coral surface for some species, especially those species that are really complex ones. Recently, a great solution has come alive, it's photogrammetry. This technique relies on alignments of photographies with a mesh constructed from coincident pixels, resulting in digital surfaces with topographical information, which therefore maintain the complexity of reef landscape for future computer analysis. It can be performed with high technology, like stereo cameras and fancy equipment. However, it can also be done very simply by recording videos swimming over the reef surface with GoPro cameras. After processing, the GoPro videos can also generate good 3D surfaces prone to have data collected outside the field on digital analysis. And besides recreating the reef surface, photogrammetry also enables to model single colonies, and this is really advantageous to monitoring corals from areas with low coral cover and when we need a detailed and accurate data collected from the colony level, not only from the reefscape. However, we still need to properly understand how 3D modeling is different from 2D photos regarding the data that these two approaches generate. To go further on that, we selected 23 colonies from an important reef builder in the South Atlantic Ocean, the massive coral Montastria cavernosa, and from each of these colonies we took photographs and recorded video with GoPro cameras. We photographed the colonies with a scale included on it and as orthogonal as possible, and the videos, besides being scaled, were recorded from around 1 to 2 minutes, and we ensured the covering of the entire colony surface. At the end, we had photos that were ready to be measured, and videos that were processed to build the digital models. So from the video generated with the GoPro cameras, we could extract around 100 frames in Free Studio software, and those frames were loaded on Metashape software to build the 3D model. To construct the model, first we had to align all the frames, and trying to find the coincident pixels is something that the software do by itself. And after that, we had a cloud of pixels, first a sparse cloud and then a dense cloud. and all those pixels were connected and created a mesh. And over this mesh, we could overlay a texture that came from the coral photos we took before. And this brings reality to our model so we can properly take the measurements we need. And then the digital model is done to be measured in other softwares. From both 2D images and 3D models, we are able to measure the colony perimeter and surface area, which are variables more related to size and growth, and also health variables like the number of bioroders, the percentage of coral border in contact with algae, which are potential competitors to corals, and also the health area of the colony. 2D images were analyzed at ImageJ software, while the 3D models were measured in MeshLab software. So we observed that perimeter, surface area, and the number of bioroders were overall smaller in the 2D images, and consider that the 3D model is a good representation closer to reality by maintaining coral complexity. 2D images actually give us underestimate of such variables. When we consider the relative variables, we observed no statistical difference between the two approaches. However, it doesn't necessarily mean that they are similarly estimating coral health or the percentage of border in contact with algae. When we take a look to the data, actually we can see that 2D images sometimes underestimate and sometimes overestimate those variables. So actually, in this case of relative variables, 2D images are giving us wrong estimates, and this is a problem, right? By using the coefficient of variance between pairs of years, we could check for temporal variation between 2D and 3D approaches, for both perimeter and surface area, which are variables not supposed to change from one year to the other, especially on massive slow-growing corals as Montastraea cavernosa. We then observed that both 2D and 3D approaches are similar as they do not differ in the interannual coefficient of variance for those variables. So these results indicate that both methods have the same associated intrinsic error. So I'd like to conclude highlighting that 2D underestimate growth variables, which despite being a problem for studies that needed accurate observation for growth rate, for example, is not a huge problem for spatial and temporal comparative studies, as the same error will be maintained across the spatial or temporal scale. But this is not the case when we consider health variables, because such variables are really important, especially with all the impacts that coral reefs are facing recently. And actually, we saw that 2D gave us wrong estimates of such variables. So it's really a problem for studies like that. And when we think about data collection, both 2D and 3D approach has similar time and logistic on the field. So in the field, it's not a huge problem. In the lab, it's a little bit different because 3D it's more time consuming, but actually the workflow is quickly automated after some runs. So even though in the lab 3D is more complicated, you can easily get used to it. And besides, only 3D models allow from among several approaches, allow us to really estimate complexity and to evaluate geometric and topographic variables, which is really important when we are talking about three-dimensional organisms as corals or the reefscape. Therefore, embracing 3D modeling to monitor coral reefs even at the colony level is not only feasible, once it's possible to perform it with cheap and easy to carry GoPro cameras, but really necessary, especially when we consider that some variables are only possible to measure in 3D models, like volume and complexity indices, for example, and the accuracy that this approach provides. Thus, even when investigating classic questions like coral growth or health, we can really benefit ourselves by using new technology. So thank you very much for your attention, and I'd also like to thank my colleagues at Marina College Lab and the agencies that provided funding and infrastructure to conduct this work. And here you can find several ways to reach me, and I'll be really glad to answer any question or to discuss interesting ideas. Thank you very much.